The purpose of this lab was to observe the effect of differing wavelengths or different colors on the rate of photosynthesis in a plant cell. The four colors that we used in the spectrum were white, red, green, and blue light, and the color was manipulated using food coloring. Also, the rate of photosynthesis was measured via the oxygen output in the leaf dots that we were using. The independent variable was the color of the light, and the dependent variable, as I said, was the oxygen production rate, because usually oxygen output in a leaf has a direct relation with the rate of photosynthesis. Our hypothesis was the most effective wavelength would be white, and then red and blue, and finally the worst would be green. Photosynthesis is a biological process for converting raw materials using light energy into chemical energy in the form of glucose. This mostly occurs in plants and algae, which are types of autotrophs, and autotrophs are organisms that produce their food without the need of living organisms, and this is a chemical equation of photosynthesis. These are the materials in our experiment. We used food coloring to represent the different wavelengths of the visible spectrum. We used red, blue, and green food coloring. For the white light, we didn't use any food coloring. The ivy leaves were used to represent plant cells. The warm LED lamps were used to represent light, because plants need light to photosynthesize. Also, sodium bicarbonate, which is basically baking soda, was used to represent carbon dioxide, which is also needed in photosynthesis. To run this experiment, we first created a 0.2% bicarbonate solution as an environment for the plant cells to photosynthesize. Secondly, we utilized a vacuum to remove all traces of oxygen from the leaf discs before we put them in the solution, because we wanted to make sure that the amount of oxygen that they started off with was constant. Third, we placed the leaf discs in the bicarbonate solution and waited for them to rise to the surface, because depending on how much light they received, depending on the wavelength that they were in, they would rise at different intervals. We ran this type of experiment 3 times because we wanted to make the results as accurate as possible. The third was the most successful experiment because we used all 4 wavelengths with 4 different light sources, and we proved that the control was the most effective, the red was the second, the blue was the third, and the green was the least effective. This is a table and the graph of our results. For the table, the 3M10S, for example, represents 3 minutes 10 seconds, and all those times refer how long that each disk took to rise to the surface. For the graph, the y-axis is the times. For example, 1912 is the same as 19 minutes 12 seconds, and the horizontal axis displays the 3 disks that rose to the surface first. Overall, we proved our hypothesis correct, that white light was the optimal wavelength. The reason for this is that with white light, a lot of different wavelengths are presented to the plant cell, and with more wavelengths, it's receiving more photons, and it needs photons to produce ATP and NADPH that create more oxygen and increase the rate of photosynthesis. Now, green light was the worst environment for the rate of photosynthesis, because since plant cells are green from chlorophyll, they reflect all green light, so they weren't getting any photons at all.